Pharmaceutical Processing, in conjunction with Interfex 2017, presents Interfex Live.
All right, so good morning again. Welcome back. We're going to be talking about the regulatory aspects of, hold on a second. Okay, there. So we're going to be talking about the regulatory aspects of continuous manufacturing. We're going to kind of step back for a little bit and try to go back over some of the criteria that's required. We have a new speaker. Bob Chu here is going to be standing in for Alice. Sorry about that. We had a last minute change. It seemed to be, I don't know if it's a spring fever thing, but we've had like last minute changes all day long, so we'll see where it goes. But I want to appreciate Bob stepping up. So having said that, we're going to be starting out. So we're going to say, okay, let's talk about continuous manufacturing. We'll drill down into specifics as we need to, okay? And the focus will be on regulations. And I don't think I have to tell you how many times this has been back and forth. And having worked for many companies myself, I can tell you it's something that nobody agrees about. And the regulations, with all due respect, are not a heck of a lot of help. But they give you a lot of leeway, which means you got to come up with the approach that's going to suit your own business. So with that, Bob, why don't we start with continuous manufacturing and what's the concept?
Well, the concept, which the regulators have recognized in many different forums. Okay.
Oh, sorry about that.
Yeah, which the regulators have recognized in many different forums, and they have this emerging technology task team. Continuous manufacturing allows you to reach a steady state and keep it that way, and manufacture ad infinitum. All right, and so the regulators and the rest of us chemical engineers recognize that once you get to that steady state, it's fairly straightforward to hold it there, as opposed to a batch. You start up, you manufacture, you shut it down, you clean it, you start it up, you manufacture, you shut it down, you clean it. So they're all on board with moving in a direction of continuous manufacturing.
So predominantly, what does the, so pretty much we've talked about several concepts here. Many of the speakers we got are going to be talking about continuous manufacturing. So where do we, what's the future up and what's the regulations? I mean, are they changing along the same lines, keeping up with the trends now? Or?
Well, let's see. So the U.S. GMPs talk about continuous manufacturing when they talk about what's a batch. So it's already covered, and we could quote it if we need to.
Sorry about that. So, is that a problem if I put this over here? Because I just realized I can't see the damn thing. Your lovely face.
Well, they can't see your lovely face.
Yeah, that's okay. You don't need to see me.
But anyways, the batch definition in 21 CFR 210.3 defines a batch as a specific quantity of a drug or other material that is intended to have uniform character and quality within specified limits and is produced according to a single manufacturing order during the same cycle of manufacture. Additionally, a lot is defined as a batch or a specific identified portion of a batch that has uniform character and quantity within specified limits, or in the case of a drug product produced by continuous process, here we go, it is a specific identified amount produced in a unit of time or quantity in a manner that assures it's having uniform character and quality within specified limits. Okay? So again, produced within a specific unit of time or quantity.
So the batch definition also covers uniformity and characteristic quality within specified limits. So what definitions are we talking about, like time specific information? I mean, this is something that's left up to the owner to come up with?
Yeah, and you could think into the future. Let's talk about a finished dosage line. All right, continuous tablet manufacturing, and Pfizer's got this going, and others do too. But you're taking the raw material, you're blending it, you're putting it through the process, and you're tableting it, okay? And then it gets filled into specific units, which are serialized. Now, those units can easily be time-stamped when they come off the line.
Okay.
And with today's manufacturing process control, you know the conditions that were occurring at each point upstream in the process as that material flowed through the process, right?
Right.
So therefore, you could basically tag that individual serialized dose with the manufacturing conditions all the way through with the right automation, data historian kind of thing.
Right.
So that would be a way of looking at how you assure that these units are of the same quality in a given period of time. That given period of time, it could be, all right, you start up, you reach steady state, now we start the lot, we run for however long, and then at some point, we stop, and there's a shutdown, right? That's your lot.
Okay, I mean, yeah, I get that part, but I think one of the things that people run into, and I know we ran into continually, is process verification. I mean, the answer is, okay, you've done that, so how do you know from time to time? So does that bring into play that the thing is continually in a state of verification or process verification?
Well, that would certainly be prudent and ideal. I mean, the chemical industry and the food industry has been doing this for decades, so we kind of know how to do this. We know how to maintain processes within a state of control. We know how to monitor them. We know how to react to perturbations in a chemical process.
Mm-hmm. So a lot of what we know from the chemical process, we also find that its controls are also applicable to the dosage form process. So I mean, I think a lot of times, where do we define, the definition of batch is how we define it. So if you decide you want to define batch as bulk, or you want to define batch as number of units, or you want to define batch as finished dosage forms, I mean, what do we usually do there?
Whatever you want to do.
I like that.
Now, the problem, of course, comes in when you have the raw materials and there's variability, right? Mm-hmm. So you need to, your process, you need to understand exactly how the variability in the raw materials affects the final product and whether or not you can adapt your process to the variability in the raw materials. All right? Because as you're doing this continuous processing, you're obviously adding raw materials, probably in bags or something like that, right?
Yep.
And putting it in and understanding how that affects the process, either physically or chemically. That's a prerequisite.
Okay. So it's also the environment. I mean, that's the other piece, right? I mean, in the case of bioprocess, it's all about the components used, that are one-time use and a single use, or as opposed to the contact surfaces. I mean, brings all this into play, but all of these things have to be continually verified. I mean, that's the important part. I mean, I think what a lot of people forget is once you're in a continuous process, allegedly you're in steady state. So perturbations become more like tidal waves rather than perturbations. And I think people understand that. They don't realize that that's the problem. And it's the starts and the stops that get you into trouble. So I think I think that's important. One is you're much better off in a steady state environment, right? And I think we should define that as, we talk about, so it's defined not only as the quality, it's defined as the commercial. And I think that's the key here. And I think that's one of the ones that people, they discuss ad infinitum, right?
Yeah, you definitely need to know how to accommodate perturbations and have procedures on how you react to the major ones, and presumably your process control would adapt to the minor ones. But one other thing is, with a batch process that you validated, you know the size of that, and all the cleaning that needs to occur, and all that kind of thing is all figured out, presumably. With a continuous process, there could be impacts of time. So you are processing this material for hours or days, and what happens inside the equipment? Does material get aggregated? Does it clump? Does it adhere? And what does that do to the process? Does it degrade? So understanding what happens inside the equipment over a period of time that you're going to process is something that you really have to know and study.
Okay.
Now,
So let's just, so the point is, in an ideal process, in an ideal continuous process, we should do what? Real-time release, real-time exchange. So in other words,
so a continuous process gives you the opportunity to more readily do real-time release, okay? Because you've got steady state conditions, you know that they're in the spec, and therefore you're able to come up with a real-time release scheme, set of parameters that are used to make that release.
okay, you got any examples or that you've worked with? I mean,
No, I don't.
okay.
Now, I've heard several case studies from Pfizer, from Lilly, et cetera. So Pfizer's got this thing going up here in Groton, Connecticut, where it's a fairly small physical scale for tablet manufacturing. And their idea is, well, we'll run our clinical batches through this, and then we will use the same equipment for our manufacturing runs. All we will do differently is the length of time of the run. So if you think about, okay, traditionally you move from clinical to commercial, you have a big scale-up factor. The only scale-up factor in what they're doing is time. And so the only thing you really need to consider is what I was talking about a few minutes ago. What's the impact on the equipment of running these materials through it over a long period of time?
Okay.
Now, Lilly looked at it from more of a traditional chemical engineering perspective. When you have hazardous hydrogenation and other types of reactions, where you have highly exothermic reactions in API manufacture, that by running a continuous process, you can generally have a much smaller volume of material in any given point in the process. And therefore, it's much easier to deal with the exothermic reaction, deal with the high pressures, deal with the hazardous hydrogen or something like that, and just handle it in a safer, more economical manner.
So, and anything there's similarities on the bioprocess side too? I mean, we've seen a couple of guys do.
I don't have personal experience
Yeah.
with the biotech. I know that several major biotech companies are working on continuous bioprocessing.
Mm-hmm.
So, I don't know, anybody in the audience want to comment on that?
There are roughly 20 licensed biopharmaceuticals made continuously. Is this the upstream?
Everybody hear that? Okay. He said there's at least 20 substances that are licensed for continuous manufacturing, at least upstream.
Okay. So there was a couple that we're going to be talking about this afternoon on continuous process as well. I mean, one of the ones that gets a lot of airplay is Janssen's on, sorry again, small molecule side. I think the other one that we're going to be talking about is some of the drug substance stuff. So what are the basic trends that we've been seeing? I mean, you know, I think we see pieces. I mean, bottom line is what's the overall trend that we've been seeing? People are switching over. I mean, where are we at?
We're not seeing a tidal wave of switchover. We're certainly seeing a lot of R&D work from big companies and smaller companies looking at this. You basically have two groups. You have the very small companies that don't care what everybody else does. They just do what makes sense. And then you have the big pharma who have their R&D groups, their new technology groups that are looking at this.
And it takes quite a while. I mean, you know, and I won't get in. We're not going to get into that story there. But so, if I was going to do this, obviously, you know, what sort of a team should I be putting together? I mean, that's key. I mean, this is something when we talked about, for example, we talked about quality by design. It was always, well, where are the skill sets?
Yeah.
To me, they seem very much the same. So why don't we talk about that a little
Sure. It's traditional chemical engineering, it's traditional statistical process control, which this industry has kind of forgotten because they're too focused on what do the regulators want to see. Right?
Absolutely.
All right?
You don't have to convince me of that.
And so to answer your question specifically, who do you want on your team? People who don't know the regulations. Who can therefore think outside the box.
Right, that was I believe there was people that we were going to have on a quality risk team. They shouldn't know anything about quality either, but yeah, okay. It seems counterintuitive, but I get it, okay. Yeah.
Because, you know, if you take people from the petrochemical, the food industry, I mean, this is old hat, okay? My mother, God rest her soul, back about 20 years ago, was working for Unilever Foods and modeled using SPC a pizza sauce process and came up with like 20 different variables and put it into this statistical process control model that was able to then drive the process, eliminate inline testing, and basically make it predictive for pizza sauce, with the variability of tomatoes as the raw materials.
I got it.
Okay?
Yeah. So there are packages that let you do this now. I mean, I think people forget this, you know. I mean, the bottom line is the first thing they do is I got to go out and get myself a PhD in statistics. It's not necessarily the truth anymore. I think you need to have an understanding of process control. But what has been your experience? I mean, when you deal with a client, what do they use? What are these packages they use? A Minitab or? Yeah.
Well, first of all, you have to get the quality unit past the point of 100% confidence.
Okay.
All right?
Yeah.
You'll never get there.
Not in this lifetime, no.
No.
Okay, yeah.
And then, secondly, you actually do have to have people who really understand statistics, because I have seen so many times when people will look at 2 and 3 sigma or 6 sigma distributions and apply the statistics as if the distribution were normal, okay? And most of the time, our distributions are not normal. Now, there's statistical techniques you can use to normalize the data, but now you need a statistician to tell you how to do that. Okay, so your run-of-the-mill engineer may not know that.
I think that's one of the problems, and what I find with a lot of cases, at least stuff that we deal with, is they take this approach where tighter is better, and they wind up having to live with this problem. And I think that's where they make their biggest mistake. You know, remember that when you said somebody doesn't know the regulations, usually they go to the, you gotta remember, we already have the goalpost set. So the question is, where do you want to operate within the goalpost? And I think in a lot of cases, they accept that. But the problem is let me rephrase it. We accept that that is going to be the limit. Not the problem with that is you're already outside the failure zone. So the point is you got to bring it in, and then work within the limits.
Well, when you say the goalposts are already set, if we're talking about developing a continuous process, then I would say the goalposts are not set.
Okay.
Other than final product characteristics, right?
All right.
The in-process limits are not set. You're going to determine those as you develop your process, as you do experimental design, as you intentionally perturbate the process to find out what happens. You need to do that. You need to do that.
I think a lot of people do just the opposite. You know what I'm saying? That's one of the things that what you learned over I've learned was, and probably learned them the hard way, was that the process is a funnel. You know, it's like this. It's not like this. And the point being is there should be varying levels, which is I'm agreeing with you. I think that in a continuous process is important, and we're going to be talking about that later today. But one of the things we found was we can measure it. The point is, does it make sense within the process? For example, you know, if you're going to make a bulk material, does that bulk material ultimately have to reflect what you want in the final dosage form, and what is that relationship? And you see what I'm saying?
Well, I will also say that traditional process control, the kind that the Japanese learned from Deming, that we then learned back from the Japanese, is not what does it take to manufacture an okay product, but get your process into control, eliminate sources of variability, and dial that in tighter and tighter until you've eliminated all possible sources of non-random variability, okay? And that's where you should be controlling your process. Now, edge of failure might be out here, but in a continuous process, and this is how the Japanese made their really good cars and all that kind of stuff, that if you remember back a few decades ago, right?
Right. They've had their events, but okay.
Yeah. But nonetheless, you eliminate sources of variability, and then you control with those sources eliminated.
Right. I think one of the biggest problems that when we talked about this the other day is the guys who are supplying this stuff. Now we heard, you know, for example, Parrish was up here the other day. We talked extensively about what they do on their end as a supply. The problem with that is not everybody does that. You know, I mean, the bottom line is the things that are going to cause you the perturbations are going to come from outside the environment. I mean, the question really is, how far back do we need to do this? How much tolerability can we build in?
So for decades, Ford had may still have their Q1 program, where they go to their suppliers and they really look really hard at how the suppliers are doing quality. You know, what does this industry do? Well, if the regulators expect it, we'll do it.
Yeah.
Is that right?
Unfortunately, that's true in most cases, yeah. But I think we're kind of outgrowing that, thanks to the guys in biotech. But yeah, in small molecule, I think we still fall into that trap, yeah.
So I was at a meeting a couple years ago of senior industry executives, VPs of engineering, manufacturing, et cetera. And the guest speaker was from the Duran Institute, okay? Global quality kind of institute. And he's talking about just how bad the pharmaceutical industry is in terms of quality. so he finishes up and he sits down. And then we moved into the section with the regulators, okay? Industry and regulators talking. And industry gets up and is talking about a QBD submission and complaining that the regulators were questioning the tint of the capsule as to whether or not it was important, okay? Now, most of us would say, oh, tint, it's not that important, right? No impact on the patient outcome, okay? Now think about a car company. Do we think that the guy in the paint shop that paints the driver's side door and then the passenger side door knows precisely how to get those tints exactly right, even though those tints have absolutely nothing to do with ability to brake, safety, fuel economy, or anything else? But do we think that that paint shop guy knows how to get those tints to match precisely? Because what happens if they don't?
Well,
You don't sell the car.
no.
Okay? And here in this industry, we're arguing with regulators over whether whether tint is important. Well, maybe it doesn't affect the patient outcome, but if we don't care about controlling tint, what else do we not care about controlling? We're getting off the subject. My fault.
That's not important. Let me just step back. One of the biggest catastrophes I've seen in project development, especially in dosage form, is there is, again, patient focus. Is, you got pharmaceutical development, you got analytical development, you got processing, you got the guys from clinic. We're talking about the classic project team cross-function. And you're trying to come up with a new drug combination, you're trying to come up with a new delivery system. And okay, regulations are out here, okay, but we're trying to come up with something unique, okay? And that's great, you go in and you do the study. And you say, okay, well, what's the risk? Right? You do the risk, you go right down the line, you do risk mitigation, risk of, we're talking early stage. The answer is the guy in medical or the guy in clinical said, well, that's not clinically significant. Now, anytime you sit at a project team and somebody says it is not clinically significant, your blood should run cold. Because that means that, you know it's 85 to 120 or 95 to 105. He ain't worried about that. These people work in the land of plus or minus 2 sigma. You know what I mean? So the point is, I think everybody has to understand what the end is. What am I trying to say? If you focus on patient safety is paramount, but the point is, if that's what you're willing to accept, the point is you have to look at that from the standpoint of what happens to the rest of the process. And in our environment, that isn't going to work. People say, well, it's got to be patient focused. I agree, but that philosophy works if you're doing a CAPA or you're doing a mitigation process. If you're developing something new, you have to look at it. Those doors have to match. That guy has to know what paint can to pull. Not only that, he has to know the number on the paint can, the spray gun he's going to use. Otherwise, in the end, it's going to be banana yellow and the other one's going to be orange. It ain't working. So what I'm saying is, I got burned by that idea. Patient focus is great, but invariably you down this road where you think you got something like this. That only works when the ship's sinking. When you're building the ship, you got to be here. And I think that's where this comes in. Continuous process, anything you want to do, have to go back. So when you do the risk assessment, you do the risk mitigation, you do risk communication, any of that stuff, you got to be very clear.
Yes, you really have to know your process, and you have to be willing to dial it in on all fronts for continuous manufacturing. But if you can do that, then you can produce high-quality material much more reliably, much more cost-effectively, much more flexibly in terms of quantity than with a batch process.
Steady state.
Steady state.
Steady state's the place to be.
Yep.
The question with that is, what's it going to cost you to get there? To me, I would say, they should all be, maybe not continuous, but they should be tunable at this stage again. We had this whole debate the other day. So let's get back to something we're talking about, process understanding. I think, we keep talking about small molecule. The guys that are sitting here, process understanding from a biotech side, that is really, think about what we're talking about, then you apply it to where you want. Process, how good is our ability to do a process understanding? That's a question. Is there anybody willing to volunteer an answer? I'm saying out there that you guys really got a problem. You guys, that's a challenge, okay? And it's more than physical chemistry, okay? And I think in the end, so what drives the process?
Well, I know that one of the major biotechs had a bad experience with disposables, in that the major supplier, remain unnamed, had variability in their materials, and it seriously affected the bioprocess. Plastic is not plastic, I guess.
Well, again, we talked about that the other day, at that point. But where I'm at is, that's only part of it.
Yeah.
Right?
That's just one example.
So, I think in a lot of cases, what I found was, and again, when we talk to people, I think you got to decide where is it that you're going to draw the line. Right? Last time I checked, we don't sell stuff in 5-gallon or 5-liter cryovac vat. We sell them as dosage forms. So we had these guys talking about pre-fill, fill finish. It's almost like we're talking in a vacuum. So when you look at a bioprocess, where do you start? Where do you start? Where do you begin? Which is it? Which end of the process do you look at? We just heard how if you start at this end, the finished system, and you work backwards. So where do you start on a bioprocess? What's your opinion on that? If you're going to do continuous, where do you define it? What's the baseline? Do you work in a vacuum? What? That's a question. Does anybody want to take a shot? Otherwise, we'll fill in the blanks. All right? Well, it's up to you now.
You got to start up back at the beginning, looking at the cell lines,
All right?
looking at the media. How's all that work together?
Mm-hmm.
And that's really hard sometimes to figure out. It's a lot harder than small molecule.
Much harder. A lot harder. I suggest that this afternoon, Dr. Muzio is going to be here. We're going to be talking about the philosophy of continuous process. Unfortunately, again, small molecule, but hey, it's going to be a great discussion. Thank you very much.
Thank you. Thanks, Russ.
Thank you.